Okay, so hopefully you watched that simple GCSE video. You watched a slightly more detailed one called from Science Sauce. Now, to stick on the C detail onto the selective reabsorption. This video is going to be fairly short. You'll be able to name the cells that make up the proximal tubule. So frequently ask you what type of cells they are, and then this is a key bit: how are they adapted for their job? So what's their job? Well, I should say selective reabsorption, where they found, proximal tubule, and then how they're adapted. So here we've got our first bit of the nephron. Remember what's going to happen. Things that are ultrafiltered come out of the glomerulus into the Bowman's capsule. They're now known as filtrate, and they start to pass along the proximal tubule. In the previous video we said as they pass along, they're being reabsorbed. And remember what they're being reabsorbed back into? Good, the vas erectus. So it'll be the blood vessel coming off here, tangles all around here to pick it all up. So let's look at the wall of the proximal tubule. Okay, so we're just looking at the cells that make up this wall. So we'll zoom away down in here, and we find these wee boys. Okay, they're known as cuboidal epithelial cells. So cuboidal just means they're more square shape than, remember, in the ileum they were columnar, so they'll expect you to say that. So this is the lumen. In other words, this is the inside of the proximal tubule. So just, I'd have some that are sitting square. We'll have a look at them. Now some things you should be able to label straight away from AS level. So they'll ask you, oh, what's this? And you'll be saying, no problems, that's a nucleus. What do you notice? There's lots of present. Yep, that would be mitochondria. So if there's a lot of them, it's there for a role. Anybody like to suggest a role? Yep. Active transport. So through aerobic respiration, they supply ATP to move molecules against the concentration gradient. So again, here's the lumen. So get our orientation right. Now, the luminal surface has these foldings of the membrane. So let me just delineate. That's one cuboidal cell. Can you remember what they're called? Microvilli. Again, that's AS. Everything here is AS. Can you think of the rule for microvilli? Increased surface area has to be. So where's the biology answer? Pardon me, voice is breaking. Microvilli. And remember way back to when I taught you that in class, having more sort of surface membrane means you could have more carrier proteins as part of that phospholipid bilayer. You're just providing more space for those carrier proteins, which are probably involved with the active transport. Now, weird thing is, this is the, if you want to say the top of the cell, this is the bottom of the cell down here, the base. This is also folded. Now sometimes they're known as basal. Now that just means the bottom of the cell. Invaginations or enfoldings. Any idea why the basement membrane would be enfolded? Once again, increased surface area. So again, you're going to have proteins here. So you're going to have carrier proteins, probably, so that would be a carrier protein. If I could spell it, yeah, it's protein. That might be for actively pump out molecules, so pump them out, or it could be for, remember this word, facilitated diffusion. So this is actually quite like the ileum. You can actively take things up. You create a high concentration, for example, of glucose, and then by diffusion it goes out. So if you had to label up adaptations, we would say microvilli. You give the whole story of increased surface area, that would be a mark. for active transport, more carrier proteins. Done. Mitochondria, make sure you say respiration. Now don't say energy. We're big boys and girls now. We say ATP. We link that into active transport. I would mention these enfoldings, more carrier proteins. So you can see that one cell potentially has, let's see if I can count, 1, 2, 3, 4, 5, 6 marks, 7. You get the surface area once, 8 marks easily. So fairly straightforward, but remember, oh, good one. Sorry, I'm not even going to bother cutting that out. Oh. So those are the little cells within that. Back here, so there's all that detail on that. I'll leave it on there, you can have a look. But look at this. These are the blood capillaries. So in here, obviously, to give you this sort of size, there's your red blood cells. So let me just remove that now. So there's a very short distance. Anything has to move out. So they give a mark for that. Okay, so leave you with that. Just log a little simple diagram for today. So we know what ultrafiltration is. You know water and ion, water's by osmosis, ions usually diffuse with the water. Glucose and amino acids by active transport, each having their own specific carrier. Enjoy. Okay, in summary very quickly. Name the cells, cuboidal epithelium. Their job, selective reabsorption. Their location, proximal tubule. Their adaptations, and we'll simply say there's A, B, C, and D. 4 that you need to talk about. Microvilli, I would give this in a bit more detail. Large number of mitochondria, basal enfoldings, and they're close to capillaries. So if you can name 4, say 1 or 2 things about each, there's easily upwards to 8 to 10 marks sitting in that. Okay, good job, all the best.